Hello, my name is Savannah Starkey, and today I will be talking about the ion torrent sequencing system for my local expert project. What is ion torrent sequencing? Ion torrent sequencing is a form of next-generation sequencing created by Thermo Fisher Scientific. It's a form of sequencing by synthesis, and it is also known as ion semiconductor sequencing because it utilizes a semiconductor chip to run sequences. The way it works is it translates chemically encoded information into digital information via the semiconductor chip. It relies on pH changes through detection of hydrogen ions being released during polymerization or DNA strand synthesis. How does the ion torrent sequencing system work? The ion torrent has microwells, and each microwell has a template DNA strand containing beads with nucleotides. It is one bead for one nucleotide. Those nucleotides are A, C, G, and T. Once each well has a bead, they are flooded with dNTPs. If that dNTP is complementary to the nucleotide on the bead in the well, from the template, it is then incorporated. Once incorporated, the complementary strand will grow. But how would you know a dNTP matched a bead's nucleotide? If the nucleotide on the bead matches the dNTP, a hydrogen ion is released. If two of the same bases on the strand are present, the voltage will double since two hydrogen ions released. The pH change is detected via the ion-sensitive field transistor under the microwell. The chemical information, like which nucleotide it was, is translated to digital format. If the nucleotide and dNTP did not match, the hydrogen ion would not have been released, and therefore a pH change would not have happened, and there would have been no voltage change. This illustration shows how the ion torrent works with the plate. The dNTP first washes over the microwell containing the bead. Let's say this bead has guanine attached to it. If the dNTP happened to be DGTP, then that would be a match to the bead in the well. Therefore, a hydrogen would be released, which causes a drop in pH. In this illustration, Q is representing temperature, and we can see that when there is a drop in pH, the temperature will increase. The sensing layer detects the temperature change, and the sensor plate will translate that into an increased voltage, and that will be sent to the column receiver. Essentially, if a nucleotide is present, a base is called, and it is added to the DNA template and incorporated into the growing strand. The hydrogen ion is released, and the ion charge changes the pH of the solution, which will cause the chemical information to be translated to digital information through voltage. How does this relate to what we're talking about in lecture? In Module 2, we've heavily focused on next-generation sequencing, which ion torrent is a form of NGS. However, I wanted to compare ion torrent sequencing to Illumina, since that's what we've been focusing on. Ion torrent sequencing and Illumina function very differently. Illumina uses fluorescent-labeled nucleotides to create sequences and captures data with a camera, whereas the ion torrent is more direct than Illumina, since it's detecting pH change and doesn't need to use a camera to record data or have fluorescent labels. A difference between the two is that Illumina has the same length sequencing reads, whereas ion torrent reads can be varying. Illumina is also able to create sequences from both ends of the fragment, whereas ion torrent cannot. It also has a higher error rate than Illumina. Another huge difference between the two sequencing systems is how the DNA is amplified. The ion torrent relies on emulsion PCR, and Illumina relies on bridge amplification. So although they are both forms of NGS, I think it's important to know the differences, because knowing the differences will allow you to choose the right system for your specific project. In addition to the weaknesses I mentioned on the previous slide, ion torrent sequencing has some other limitations as well. The first limitation is if homopolymers are present. With ion torrent, a homopolymer is more than 10 of the same bases in a row. If a homopolymer is present, the accuracy of the read will diminish. But how would you know you have a homopolymer in your sequence? A good indicator would be seeing a very large signal because the detected voltage level should reflect the length of the homopolymer. Another limitation you could run into is bias. Since this is a Thermo Fisher Scientific product, it is important to use the corresponding kits when running your PCR prior to running the sequence. If you decide to not use the Ion Chef system from Thermo Fisher, your whole sequence will rely on you knowing how to do emulsion PCR correctly. Emulsion PCR is often used in NGS systems, and it is when you dilute and compartmentalize template molecules in water droplets in a water and oil emulsion. If you do this yourself, it can introduce bias, which would be a limitation. Therefore, it's always wise to invest in the kit. However, investing in the Ion Chef kit could be a limitation for some labs depending on budgets because the instrument can cost up to $65,000. The last limitation that exists with ion torrent sequencing is the short read length. It can only read 200 to 400 base pairs currently. The throughput is also much lower compared to other methods you could use. It can only generate 50 gigabases. That means if you're hoping to sequence whole genomes, this may not be the best option. However, they are trying to improve this, so in the coming years, it may not be as much of a limitation. With what seems to be so many limitations, why would you want to pick this over something else like Illumina? Well, one advantage is that it's a direct system. There is no need for a camera or fluorescent labels. The nucleotide is directly integrated and converted to voltage. This means sequencing is occurring in real time. Overall, it is a more affordable technology and method as well. This means labs will be able to cut down their costs. Currently, Illumina costs $128,000, whereas Ion Torrent is $80,000. However, you also have to be thinking about investing in Ion Chef, so to some this could be a disadvantage. However, on the bright side, compared to other methods, the cost per run is much lower at only $1,000 per run. So overall, I think the cost for the entire system is an advantage in the long run. The third advantage is the runtime is very quick. So if you're looking for speed, this may be a good option for you. It takes 2 hours to create a sequence with the Ion Torrent, whereas Illumina takes around 27 hours. What are some controls you need to be aware of when doing Ion Torrent sequencing? The first thing you want to be sure of before running the Ion Torrent is that you amplified your DNA through emulsion PCR and not another form of PCR. If you use bridge amplification like we do in Illumina, then your Ion Torrent sequence will not work. When using Ion Torrent sequencing from Thermo Fisher Scientific, you need to buy their corresponding control kit. The control kit provides library preparation and template controls for use with each specific machine. The Ion Torrent S5 machine, for example, needs CEPH genomic DNA controls for 3 library preps, a 200 base pair human CEPH control library, and a 400 base pair E. coli, which can prep up to 10 templates. So now that we've talked about what Ion Torrent sequencing is, I am going to show you 3 different figures from 3 different papers where researchers utilized Ion Torrent sequencing. This figure comes from a paper where researchers are using the Ion Torrent system for routine identification of clinically relevant mutations in colorectal cancer. The researchers wanted to compare Sanger sequencing to the Ion Torrent sequencing to test for colon and lung cancer mutants. And then they took their voltage data and translated it into graph data. Sanger sequencing is currently widely used to look for genotypes like KRAS and BRAF in colorectal patients. So in this paper, the researchers verified the ion torrent panel and applied it to specimens using Sanger as a reference. The results were that the ion torrent could detect mutant levels at 5% and classify control tissues and detect mutant alleles that Sanger missed. So this figure is showing those results. Figure A shows the loading density of the torrent and performance parameters. Figure B is showing results of the ion torrent sequencing run that had 316 chips. And then the figure is also showing that there was a low frequency of the Cras mutant allele. Overall, these results show that the ion torrent sequencing was somewhat better at detecting mutant alleles than Sanger sequencing in colorectal patients. This paper was looking at bacterial community shifts in treated periodontitis patients through ion torrent 16S rRNA gene amplicon sequencing. This table is a summary of the sequence process. In the gene amplicon sequence, they targeted the V6 hypervariable region, which corresponded to positions 984 to 1047 in E. coli. And there was a total of over 4 million sequence reads per sample and 314 chips on the ion torrent machine. There were around 8% of amplification primers in the sequences which were removed and not included in the data. And they wanted to compare these reads to see if it would be useful to use as a way to detect certain pathogens when it comes to periodontitis. And this data shows that using the ion torrent would be beneficial in routine diagnostics because it was able to detect select pathogens. This last paper evaluated ion torrent for thalassemia diagnosis compared to traditional NGS methods like Sanger. This table is showing calculations of the ion torrent sensitivity and specificity. Overall, this data is showing that the ion torrent sequencer is successful in detecting large deletions and non-deletional defects in thalassemia with high specificity. It was consistent with Sanger sequencing, and overall they found that with the ion torrent's accuracy, this would be a decent option to aid in diagnosis thanks to its efficiency. And here are my citations. Thank you.